Hello, check.
So I would like to welcome one and all for this week's colloquium. And I just have an announcement that this week's colloquium was originally scheduled to be given by Professor Anando Shen from Jamia Millia, but due to some personal reasons, he had to cancel it. And whenever such a situation happens, I typically rely on my own colleagues inside the institute. And I keep, of course, asking if somebody can fill in. And I was very happy that Devashish actually helped me out here. So thank you, Devashish, for filling that colloquium slot, because it always is a precious slot. And he gave a title which I think is so appropriate for me to say to all of us. We need to talk, all the faculties together in one place. We need to talk and attend colloquium. We should listen to each other. So I think it's very appropriate. Of course, we are going to talk about here about how cells communicate, but we should also sort of use colloquium as a platform to talk to each other. So coming to Devashish, Devashish, of course, currently is a reader in the Department of Biological Sciences, and he joined in September 2019. So it's almost close to 5 years now. He has been in the institute. Devashish actually gave a colloquium formally in 2021 when his first paper from the lab came out in PNAS, and he shared that excitement at that time. And I think another big paper has come out, so we will hear that today. But just to give a little bit of a background for Devashish, Devashish did his bachelor's in chemistry with chemistry honors from Ramakrishna Mission, Narendrapur, and then did his master's from University of Kolkata. And then he went on to do his PhD in University of Kolkata with Professor Chanchal Dasgupta. Then he went on to do his postdoc in two places. First, in University of California, Berkeley, with Brian Krantz, who actually subsequently moved to University of Maryland. There he was there for at least 3 years, I guess, and then he moved to eventually being a research associate with Ed Chapman in HHMI and University of Wisconsin. He was an HHMI fellow in University of Wisconsin. So in 2019, he came back, and we are lucky to have him. And Devashish, thank you very much for doing this. So please, the floor is all yours.
Okay. Thanks, Jyotishmal, for this kind introduction, and thank you for giving me this opportunity to present some of our work in today's colloquium. So, I'll be talking today some of the works that we are doing in understanding how cell-to-cell communication happens. The broad perspective of it is obviously how the overall body function of overall human body function gets synchronized, and this is highly important in the context when there is a dysregulation of that happens, which ultimately leads to many different human diseases. Now, if I zoom in here and then try to see what really happens in the molecular and cellular level, what really happens is some chemicals, these red. Oh, I should put this. Okay, these red balls here, basically some chemical messengers. They release from one cell, and sometimes they interact with the same cell, or they interact with some neighboring cells. And these are a couple of different examples how this type of interaction happens. Like say in this case, this interacts with the same cell. Like here, if the two cells come very close to each other, and this same thing can happen through a gap junction like structure. And then here, say for example, if the cells are to some extent apart from each other, the similar kind of phenomena can happen. And in endocrine cell, like in hormone secretion, what happens is cells are really far apart, and then these chemical messengers actually come out of cell, and through the circulatory system, it goes and interact with the target cells. So that is now one thing you will see, like as we go from top to bottom, essentially the distance between the two cells increases. But even then, I mean, this is the fundamental mechanism how one cell talk to each other, that you have this chemical messenger release from one cell and interact with the another cell in a very simple term. I would say in that way. Now, so here I will now this is a cartoon, and here I will show real examples of few examples of the cells where this has been studied quite extensively. One is of course neurons, and then here this process has been people have studied this in quite extensively. It's a nice confocal image of neurons where you can actually see one neuron connect with another neuron. And here, probably many of you heard about neurotransmitters, and these are essentially some chemicals that gets released from one neuron and then get interact with its another neurons, and that's how these one neuron and another neuron talk to each other chemically. And now I show you, this is a pancreatic cell. This is a pancreas histology. Essentially, pancreatic beta. So you heard about insulin. I mean, in terms of in the context of diabetes, it's also acts as a chemical messenger. They release from pancreatic beta cell and go and interact with the neighboring target cell, and that's how the two cells talk to each other. That's another example. Obviously, these chemical messengers have their it is varied. I mean, it's varies in size, varies in chemical properties, varies in a lot of different things. But in overall, they do the similar job. So now this is a another example of the cancerous beta cell, where I specifically chose this example to show these exosomes. These are relatively different structure. These are kind of a large vesicle-like structure. Within it, you have a couple of chemical different types of molecules entrapped, and they by themselves whole as a whole vesicle, they actually come out of the cell and then kind of do the similar kind of job. So now, while doing this studies, I mean, this is a very classical study by John Heuser, done in a long back in 1980s. And he's fantastic cell biologist as well as electron microscopist. So what he showed is that while people are doing while cells are actually doing this thing, he showed it in mast cell that this is the one kind of secretory granule, that is what they call, actually going and is and fusing with the plasma membrane of the cell, and they're actually releasing this content outside the cell. He could actually show it at that time. And that was one example. And then obviously it followed many other examples of the many other cell types. People have shown similar kind of images. Yeah.
Next part. This thing is inside and then it releases outside.
Yes.
Is that this
precisely.
granule?
Granule, that is what they used to call it. Okay. That's the term they use for the mast cell. And these are essentially histamine that are releasing from the granules. Obviously, it was not that easy. I mean, you have to quick freeze it because this is a dynamic process going on. So you have to quick freeze it and do it. It was a little challenging at that time. Yes, precisely. Yes. And this is relatively much recent paper where this cryo-electron tomography. And this is one example I picked, but there are many such studies are going on now where people are trying to look at it in neurons, because neuron is the obvious place, as I said, well studies and so on. So there also you see a lot of these vesicles. Obviously, there is another fundamental difference I tell you here is the vesicle size. Essentially, and I'm coming into the context why the vesicle is coming, lipid vesicles, but essentially these chemicals are entrapped, chemical messengers are entrapped inside the lipid vesicles. So the vesicle size varies in neurons to other cell types significantly. In case of neurons, the vesicle size is like a diameter around 40 to 50 nanometer. But in other cell types, it can go in say micron, and that is one of the limitations. You can do only certain things with neurons, but you can actually do, like say in this case, since it was like a huge vesicle, then it was easy to image in that way. Okay, so like with this background, so essentially, what I'm talking about is some secreting Oh, again. So essentially, I'm talking about the secretory cells. They release these cargo molecules, and it is going and interacting with the neighboring cell. And these are a couple of examples of these chemical messengers. Now, what is important is what I would like to emphasize is that because the cell to cell communication I mean the is mediated through these chemical messengers, the amount of these chemical messengers released from one cell has immense importance on the strength of this interaction. And entire how this physiology works depends on one of the factors depends on this, the amount of cargo that is actually coming out from one cell. Now, and whenever there is a problem in that say too little or too more secretion occurs, that results in all sort of different human diseases. We can imagine, I mean, human disease then like ranges from say anything related to neurodegenerative, I mean, related to brain disorders or related to say immune cells, like say they also secrete cytokines. I mean, for the bug that comes in and you want to actually and then also from the hormone releasing part. So like it's a diverse effect on variety of different cell types. Okay, so now the question comes is when that abnormality happens and when the amount of chemical messenger secretion gets altered, is can we think of like reprogramming cell back to its normal? That is the question. Now, if I have that kind of aim, now what are the things that we need to know in order to actually address this thing and proceed further? Now, three things I would like to mention here is one, yes.
If the information that it's transporting depends on the amount of the thing that is getting secreted.
Absolutely, absolutely. The information, I'll tell you precisely how it is, like if say for example, I'll come into that. Maybe at that time I will, but it depends on this, like how the amount of chemical messengers, how it is released, or it actually leads to how it excites or how it interacts with the neighboring cell, and how much there are like so for example, it is prominent examples in neurons, how they actually excite the neighboring neurons. If you have less amount release, that will have some impact, or if it is more, that will have some other impact on the cell. So precise timing and the amount of it release has an immense importance in how this
That part is okay. What I was asking is that like say for example, if you have X amount gets released, say it transports certain information, which is something say X1, but if it is Y amount, is it Y1? I mean, it's a
yes, it is Y1. It's a different one. Yeah, it's a different information. Yes. In terms of chemicals, the same vesicle, there are heterogeneity non-level. So in terms of chemical, the same vesicle can have different chemical messengers entrapped into it. But it is also in most cases, in one vesicle you do see, or in many cases, in these vesicles you have one type of chemical messengers. But what I'm really emphasizing on is the amount of that chemical messengers while it is releasing, like less or more.
So if you have an exosome, if you have two exosomes, both have same information, when they're received by the next cell, do they respond similarly always?
Not necessarily. There can be varied response. Okay, so basically the three things that one would need to at least what we are trying to understand in order to address this programming cellular secretion. One is trying to understand the mechanism of secretion by itself. You have to know it in great details in order to kind of modulate it, number one. And number two is identify some small molecule in modulators. If that actually work there at that juncture, can modulate the secretion. I will come into that in my talk at the end. And obviously, if we can put it inside the cell and then see if that works to really alter the secretion. Now in today mostly I'll be focusing on the initial part where we are trying to understand the mechanism of secretion by itself. Now coming in a very simple term, if I have to say how the secretion occurs, as I said, that these chemical messengers are entrapped in vesicles, and these vesicles actually fuse with the plasma membrane, and that's how they release this cargo molecule outside the cell. Now, of course, you can imagine. I mean, you can imagine what I'm talking about is actually fusion of two same charged species, which is otherwise energetically not favorable. Now, unless you catalyze this reaction, and in this particular case, this catalysis is done by a special class of protein which was discovered by James Rothman, for which he got Nobel Prize, are called snare proteins. Now, these snare proteins, one class of the snare protein which is present in vesicle, we call that as V snare. Another is present in target membrane, we call that as T snares. They have specific intrinsically disordered regions. That means those regions where they interact with each other are otherwise doesn't have any structure, but when they come close to each other, they form a very stable structure as shown here. This is a cartoon anyway. So these are so stable that you have to boil it in order to actually heat it, boil it in order to kind of bring that apart. That is the driving force for the two this same charge species, like the two membrane to come close together and fuse. Now, in this fusion process, what happens is a very crucial kinetic intermediate forms, which we call that as fusion pore. Now, this pore is first aqueous connection between the lumen of vesicles and the extracellular space, through which all these chemical messengers actually release outside the cellular environment and does all the job. So that is okay. So now if you think that snares by themselves are sufficient to form the they indeed can form the fusion pore, but if inside the cell they are the only players that actually regulate the pore properties and so on, then you will always have membrane fusion going on and release of cargo molecules in an uncontrolled way. You don't want that, because you want some signal to be transmitted with the response of certain stimulus, say for example. So how that is controlled, and that is precisely where we are actually working on. We are trying to understand how variety of different proteins as well as membrane lipid work at this juncture and actually regulate this work with snares and lipids and regulate this entire process, and how they actually and then regulate the fusion pore dynamics and so on. And there are a few examples are shown here. Okay, so now then you might wonder that, okay, so why fusion pore was kind of, I showed you the 1980s John Heuser, that classical paper, and there onward, I mean, it was followed by many other work. Now then the question is why it is relevant even now in 2024. The point is, even if it is such an important crucial structure through which all the chemical messengers, most of the chemical messengers released from cell, we don't know the structure and structure of this fusion pore yet. The simple reason is you probably have heard about channels and pores in the plasma membrane, and in many different occasions, like ion channels and so on, or protein transport channels and so on. All these channels and pores, whatever it is present in the plasma membrane, most of the occasions they are formed by proteins which has a defined structure. So you can most of the cases the structure is known. It has a defined diameter, unlike fusion pore, which does not have any defined diameter. It is a transient in nature, in the sense that as soon as it forms within a millimicrosecond time scale, that is in neurons, in more or less in the second time scale in other cell types, it either fully fuses with the plasma membrane or it closes. That dynamicity made it so tricky to study its structure, and then structure function relationship. And we call that, these are the different modes how the chemicals actually release, and we call it as either Crescendon exocytosis or full fusion. So now the question is what we know. We know in great details the structures of snares without any, some cases with lipid membrane or some cases without lipid membrane. This is a nice crystallographic structure, and this is a very nice cryo-electron microscopy structure of snares that are actually the proteins that are involved here. And we know precisely how they interact with each other, and then what are the residues that are interacting with each other. They're highly conserved in across different domains of life. Now this is the structure information, and what we also know and what people have done is some simulation based study where they could actually simulate, and this is a coarse grain simulation where they could actually see the fusion pore formation and its structure, how the lipids and proteins are oriented in that. And this is one of the examples, one of the very few examples that we have where fusion pore people try to actually locate the dynamicity of this fusion pore within a living cell. And here they labeled the inner plasma membrane with some dye and the content that is being released with some other dye, and using microscopy they could actually see the dynamicity of the pore to some extent. But still the problem remains that we don't know, that there is no technology till now where you can to see the fusion pore dynamics within the cell in such a great detail that we can actually say, okay, these are my parameters that will tell me the amount of cargo release that I'm talking about, which will have impact on all the things that I've been talking about, right? So now that is one. So yeah, so in order to do those things, what I'll be telling you today is the effect of few proteins as well as some one lipid component, and our approach is essentially a kind of a bottom-up approach. So we use reconstitution technique. We try to recreate this entire thing in test tube essentially, and we try to then slowly bring the complexity into it, and how then at the test, and then finally test is inside the cell some of the phenomena that we observe. So now, so the point is, so that means what we do. So our tools is essentially we do a lot of in vitro biochemistry, and there one reagent that we use is called nanodisk. Essentially, you can imagine that you will need lipid membranes. You will need to incorporate protein in it because we are studying membrane fusion. So the nanodisk serves as one of those model membranes. The advantage of nanodisk is essentially it's a flat bilayer disc like structure you have, and where it is surrounded by another protein, and the advantage of this is you can actually control the number of copies of the protein that you're incorporating into it, which has immense importance because in real scenario in synaptic vesicle these snares are present in multiple copies. So in order to know the effect of those why there is so many copies, whereas few of them are important is sufficient to elicit membrane fusion, and those kind of question you can actually ask. So that's why it is important. And not only that, people use nanodisk for a lot of structural studies as well, because you have access, unlike liposome which is enclosed thing, you have access to both the sides here. So you can do a lot of different perturbations. So hence people use nanodisk in that way. Now we can make, I'll tell you our advantage why you use nanodisk, and we use the nanodisk that we make. We can make, like say, around from 13 nanometer approximately to around 50 nanometer diameter of the nanodisk, so that you understand the scale that we are talking about. Okay, so now what we do is we incorporate V snares in nanodisk and T snares in another acceptor membrane. Now that membrane can be a liposome or can be another membrane, black lipid membrane, on that I'm coming in a minute. So and then you allow these two to react to each other first in absence of any regulators. What happens is because of this membrane boundary condition, as I said you that this fusion put is kind of it doesn't as soon as it forms it fully fuses. So if you use two liposomes to each other, so it will form fusion pore, but it will suddenly make one lipose. So you won't be able to kind of trap the pore. So our idea is to trap the pore and study the pore properties. So this is one of the ways you can actually trap the pore in its non-dilated conformation. That is the another advantage why we use nanodiscs for our assays. So then now then what we have done is we have done one assay where we entrapped glutamate inside our T-snare vesicles, and we allowed the V-snare nanodisc to react with it. Glutamate comes out through the pore, and in the medium we had glutamate fluorescence sensor. Essentially, these just interact with glutamate and lights up, and you basically measure that in the fluorescence and as a function of time. So when you did those experiments, and we did this, this is kind of the graph that you get percentage glutamate release, and obviously the percent is not that much because we don't have that much regulators added here. So you get more or less this, this is a red curve, shows that when you have PE, PC, PS, these are the like phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine. This combination is the lipid constituent of my in this case liposome. So now, interesting observation what we got is that the moment we added another lipid, which is diacylglycerol, we got a significant reduction in the percent glutamate release. Now, this diacylglycerol, and I have another thing here, PIP2, but believe me, we have done experiment with. I don't have the data here with only DAG. We have even titrated DAG as well and got a kind of a really graded response. That yeah.
In this liposome that you are talking about, this I see that you have dumbbell kind of two things and connected like that. Is it that they are connected by some Van der Waals spores or something like that? Or
no, it is. They have a
independent. No, in that picture,
they have transmembrane domain. The snares. They have transmembrane. You are talking about these, right?
Yeah, each individual thing, how they are connected. I mean, what kind of force? Is it some weak Van der Waals force kind, or
they are hydrophobic in nature, so they go inserted inside the lipid membrane.
Are you talking about the proteins or the membranes talking to each other?
This is
this each unit looks to be independent, individual units, and they are just arranged in some way.
These are
what kind of force in between them? That was the thing.
Hydrophobic interactions. Yeah.
Some hydrogen bond. I was just thinking like what kind of bonds are there, which gets open up when you have this secretion or something.
Yeah. So these are hydrophilic tails, and these are hydrophobic. Yeah. Yeah, essentially these are the proteins which also have the kind of hydrophobic characteristic that actually gets into the plasma membrane. So and this in the lipid you have one component which is diacylglycerol that actually reduced this secretion significantly. Now this was kind of a serendipitous discovery for us, and then what we did is we used one of the proteins that we wanted to test. That is MUNC protein, MUNC13, which is one of the regulatory proteins that I just mentioned before, which is known to interact with this diacylglycerol. When we added this protein at the beginning, we saw that now this it is showing some increased secretion. As you can see, the glutamate release has increased. There is another protein, MUNC18. We also saw that also showed the similar increase. But interestingly, this MUNC18 protein does not have any lipid binding domain. It interacts with only snares, but still we could actually see this decrease. And these are a few parameters that are quantified here. Now the question is, what does it tell you about the pore properties? Because these are ensemble assays where it is, the glutamate release is being measured. It can be that these factors are actually acting in many other steps in the steps before the pore formation. So essentially you need an assay to say that okay, this is my assay where I can say that this thing happening. Hence, I can tell that the fusion pore has opened, and that's why whatever readout I'm getting is for the pore. And if I don't get that readout, I will say no, there is no pore opening. So for that, and in order to get more deep into it, now we do a single in a single in a single pore level, in a single molecule level, we study that fusion pore properties using this particular assay, where we basically have now nanodisk containing V snare as our donor membrane, but acceptor membrane is a black lipid membrane. What is that? Essentially, you have two chambers, cis and trans chamber, which is separated by a small pinhole of 150 micron diameter. You basically paint lipid there, and there are ways you have two electrodes connecting the two chambers, and by measuring capacitance, you can actually tell how good is the membrane. So I'm not going into too much detail of the technique, but after measuring the capacitance, after you know you have a good membrane there are ways you can first introduce T snares into that black lipid membrane, and then obviously you can actually use many different types of lipids for your choice to make this membrane, black lipid membrane, and then you first introduce T snares. It goes into the black lipid membrane, and then you allow the nanodisk containing V snares. When the pore forms, you get a current, and that current you measure as a function of time. The current is essentially the picoampere current I'm talking about. So that is all the assay that the technique is. And when you don't have any snare there, V snare in the nanodisk, you get a baseline current. You don't this is where you have say V snares, and you get a real fusion pore which is trapped in its as I said, you kind of a metastable conformation. And this is the single pore properties that you are actually measuring.
Just a quick question. I need to go back one slide on the device, the measurement device.
Yeah.
So you have a membrane separating two different parts of liquid, right? So what you're saying is you have unless and until the membrane pore opens, there won't be any exchange of ions or something,
Absolutely.
right?
We have potassium chloride.
Potassium chloride.
Asymmetric potassium chloride.
Right. So then this equalization will lead to some current. The current will lead to some equalization of ionic strengths, right? So it's the ionic current that you are measuring, and how you are measuring that, the ionic current. You're just having some voltage measurement? Or?
So basically you can keep it in a zero voltage, but because we have an asymmetric, solid.
Already there is a potential, EMF.
Exactly. Using that, you can actually measure the current.
Okay, so change in EMF. Okay, fine.
Or you can also apply voltage.
Or you can apply.
You can apply voltage.
Fine.
Both those. So now this is a typical, single pore that you get. This is the closed state of the pore. This is the open state of the pore, and there are three epochs are shown for a long, fusion pore recordings. Now here the lipid were like P, PC, PS, but the moment again when you added this DAG, you can actually see this is the otherwise closed state of the pore, open state of the pore. Here you see it got fully flipped. This is pore is now mostly in its closed conformation, but rarely going into the open conformation. That probably explains why in the previous ensemble assay we got reduced glutamate release. And these are the few measurements that you can actually do. These are the current measurements, open and closed states, histograms. And what you also can measure is essentially the kinetics. That means the time the pore stays in its open as well as closed conformation. And that will tell you the essentially the dwell time, open dwell time and closed dwell times. So as you can see here, the diacyl glycerol is significantly reducing the open dwell time of the pore. And then what we did is we added Monk 13, which I told you that's one of the regulators which interact with DAG. sorry, at the beginning, and it actually opened the port in its relatively stable open configuration. It is stable open now. But another interesting thing we observed is that once we opened, it opened in a two different open conformation, as if it opened and it is trying to kind of dilate essentially. So all this information you can actually get from here. That would, yes. BLM.
What happens if you switch it?
We have not.
So if you put DAG on the nano disk, do you think it's going to act the same way?
We have not done that yet. Yeah, we have to do that. But yeah, it is in this case. It is in the, and then it is actually in the plasma membrane also. It in the cell is present in the plasma membrane as well. So it's kind of. But we will do that.
Okay, another question I have is, what is the resolution on the time resolution?
1 microsecond. Yeah. So now that suggests that this MUNC13 protein is interacting with the DAG, and most likely actually relieving the clamp that is actually mediated by this DAG. And another so now here I would emphasize on the two parameters that we are quantifying again is, one is conductance, as I told you that we see like two different open states. Actually, this conductance tells you the physical size of the pore. And this is another thing, is the closed and open dwell times. It tells you the kinetics of the pore. That means one is the size, another is the time at which the pore stays in that particular conformation. Both these properties will have impact on how much amount of cargo it is releasing. Probably it is getting released from the pore, through the pore. Now, in order to understand whether this is really what we are observing really going through the DAG mediated pathway, this MUNC13 interaction that I'm talking about, and on top of that, another parameter is we wanted to actually vary these two things. Like one is the size, and then another is the kinetics of the pore. And then in the remaining talks, I will actually show you a couple of examples how you can actually vary these two parameters. That is essentially what I would like to emphasize here. So in this case, what we have done is we have changed one amino acids in MUNC13, which people knew that it interacts with DAG. And now what you see is that you see that much longer closures of the pore are appearing. Number one, that means it is altering the kinetics, and this is actually very interesting observation. Number two is the size gets significantly increased. That means in its normal wild type condition, MUNC13 is relieving the DAG mediated clamp, but it is not actually fully, it is kind of restricting the pore in a kind of a narrow conformation, although it is kinetically stable. So this kind of information we can actually get from this study. So why this is important? It is important because if a narrow pore stays for a longer time in its narrow conformation, is equally good in releasing the similar amount of cargo if the pore dilates in a fast time scale. So basically, these are the two things that are actually might have the similar effect if it stays longer in its narrow conformation. That is exactly what I'm kind of talking about. What are the factors that actually regulate these two things, and can we regulate these two parameters as per our need? Because we have this assay that we can actually test. Okay, so then I'll talk about one other protein, which is that
No idea right now of the diameter of the pore. Narrow
Yes.
means it's just
Yes,
narrow
it is
with
around
respect to some conductance.
3 to
right?
4 nanometer. You can measure it.
How
In
do you-
this case, you can measure it. The conductance has a relationship with the diameter. I mean,
Okay.
yeah, you can measure it. It varies. It varies from say 2 nanometer to 4 nanometer under a variety of different conditions. Of course, I mean, you have to keep in mind that in real scenario, it is a dynamic structure. You have to keep that in mind.
I think I was always What is that controls the time, I mean, the dynamics of that pore?
Yeah.
When it's,
Yeah.
so what is that tuning then?
I believe that it has something to do with the proteins and the lipids. The proteins that are actually going, and if you see this structure, this structure, you have these snares, and that is what actually holding the pore.
That's true, but,
And when these regulators come, they interact with it here. They can't really access this, but they regulate here. And we now we are kind of writing a paper where we are showing that these and these are allosterically linked. Hence, because of that interaction, this actually regulate. So it's a transnare complex. It's widths like this. I told you that is a very strong complex, but the state that we are trapping the pore, it's rather dynamic. This complex is rather dynamic. We have proof of that. That these proteins can actually go or intercalate there. And can actually dynamically control these properties.
is it the time scale depends on the size of these also?
Possible. Possible. That is the one thing we don't know. Possible. Because what happens in neuron, it is way faster than in other cells. It's possible. Absolutely possible. Yeah. Now, this is another protein, which is MUNC18. This, as I mentioned you briefly, that it doesn't have any lipid binding domain, it interacts with snares. Now, it is a very important protein for cell, and you cannot delete it. It's an embryonic lethal, and only in one organism, at least I know of, in worms, you can delete it, but it basically paralyze the animal. But it is why it is so important, because even one single mutation of this protein can lead to variety of different diseases, like in all the epilepsy that you hear of, basically because of this particular protein, and not only that, other non-epileptic disorders as well as caused by this protein. So when we added this particular protein at the beginning, again, we saw it could also relieve that DAG mediated clamp, but you see two things. One is the pore's conductance is larger when it is relieving than MUNC13 mediated pores that I was actually talking about before. Number one. Number two, it is actually showing some relatively longer closures that we observed here, which was not present in case of MUNC13. So essentially, this is a protein that has that although it relieves the DAG mediated clamp, but differentially affecting these two properties that I was talking about, the kinetics and the size of the pore. Now, then what we did is, in cell, obviously you have both present, right? So what happens when both of them are present there, and what sequence they act on the pore? So at the beginning we added MUNC18, and as I said you that it is kinetically less stable. You can actually see longer closures, but size wise, like diameter wise, it is larger. And on that pores we now added MUNC13. And it turned out then it kinetically stabilized the pore. But MUNC13 cannot change the diameter. It kinetically stabilized it in its open conformation. Then we did the reverse. We added MUNC13 first. It stabilized the pore in its open conformation, but size was small. And then when we added MUNC18, it didn't have any effect. Rather, like neither it increased the size of the pore nor it kinetically alters the pore. So it gave us some indication that if you want to dilate the pore, that is the natural tendency of the fusion pore, what will be the order of reaction of these two proteins? But also these two proteins can independently act on the pore inside the cell. Not necessarily they have to be coupled. The point is they differentially regulate these pore properties. So then what we did is we added them together also, and we found that in the size of the pore is larger as well as the kinetical stable, as expected when you add both of them together. Okay, so now I'll now switch the gear, and so essentially the point of showing all this was to actually emphasize that how these proteins and lipid modulate these two properties that I mentioned. Now I'll switch the gear and show you another protein that we we have worked on, and we have seen that this protein also can alter the pore properties. This is alpha-synuclein. This alpha-synuclein is a highly abundant protein in neurons, and it is 1% of the total brain protein. It's a so important protein, but unfortunately we don't know the function of the protein yet. And why it is even more interesting to work on is its obvious connection with the Parkinson's disease. Its mutation is what actually causes oligomer formation, or insoluble aggregates of this particular protein is observed in all the Parkinson's patients that that is one hallmark. And in parallel, that results in abnormal secretion from neurons. That is essentially what you get in Parkinson's disease. So when we added alpha-synuclein in our assay, we also observed that it also alters the kinetics of the fusion pore, but it does not alter the size of the pore when you have only snares forming the fusion pores. So as you can see, with a concentration dependent manner, sorry, it is increasing the fraction of time close. That means the time in which say for example 30 minutes recording, what is the time in which the pore is even in closed state and open state, you measure that, and kind of from there you get the fraction of time closed. Okay, so now with this information, the question is, can we go towards programming the cellular secretion? Now obviously we are working on trying to understand, I don't have time to tell all the stories. We are also working on trying to understand the role of calcium sensors, trying to understand the role of in many other regulatory proteins, as well as snares by themselves. Now in parallel, are we in a position to start this this project of programming cellular secretion? What I mean by that. So I'll give one example what I mean by that. So this is a MUNC18, the protein that I mentioned, that is so important, but you cannot delete, but you can delete from worms. So this study, what people have done, they deleted from worm, and then they did a rescue experiment with wild type and one more mutation, mutated version of that protein, and it turned out that that particular mutation is actually better than the wild type. And it's not that all the time you mutate and you see kind of reduction in the protein. It's better than that. That's what you call it as a gain of function mutation. And what they measured is essentially the movement of the worm in a liquid medium. And it turned out that the movement of the worm is significantly enhanced in that mutated protein. Right now, the question is, what happens if I put it in our assay system, and then what happens when we did that? It turned out that this mutation actually reduced the open lifetime significantly. What is kinetically, if you remember, MUNC18 was kind of not kinetically that stable. This is pink one, the wild type. But here, this is the mutation. You can actually see that close dwell time shifts significantly to the left. So it actually stabilizes looks like it is kind of allowing the pore to stabilize more in its open conformation and allows the more and more cargo to release. Most likely, I cannot say for certain that most likely that would explain this kind of cellular phenomena. Now the question is, if I have this phenotype and I have this information, this is a nice structure which is available, and this nice structure tells you the specific regions of the MUNC18 that is interacting with snares. Now can we use this information to design some special class of peptides called stapled peptides? Probably many of you heard about. Stapled peptide is essentially small peptides that are locked in its alpha helical conformation with some stapling, and these stapled peptides can it go and compete with the existing interaction that is actually going on and part of this?
Quick question. So in that mutant D326K that you have rescued the function, right? And it is even better in the sense it's gain of function mutation, but organism level, right? That some movement parameter, right?
Yeah.
How does that get connected across events of all the sorts that it is controlling?
Yeah, that's one of the things that we would like to know.
Okay.
But one
It's
can
just a fusion thing,
Yeah.
and that is controlling the
but people
movement.
have done extensive studies on that particular mutation because of its interaction importance, essentially because it's gain of function mutation. People have done strategic studies in order to actually know what are the things that it is doing, but most of the studies are done in vitro. And very few studies, because you cannot delete monkey, and the only model system is this one.
This one. Okay.
Yeah, you can actually work with this mutation. So now this is a staple peptide that we are trying to design. And the first step, I mean, obviously there are multiple steps, multiple hurdles that one has to actually face in order to design such peptides. Essentially, we have to first decide where to design those peptide, and its binding affinity, its stability, and obviously at the end its cell permeability. It has to go inside the cell and does its job, right? So, but the very first thing we test is we design the peptides and try to do some simulation study and see if it actually makes sense to order those peptides, expensive peptides, right? So I mean, Shahina in our lab has started doing some of the simulation studies. Don't ask me too many questions about simulation. We're kind of learning it. We are kind of doing these things and see that these are the two peptides that are based on snares that we have seen interacting with snares so far. And we have this, we are collaborating with Kalyan in TIFR Hyderabad to get those peptides soon and test it. And so obviously these are all the things that one has to actually follow in order to design these peptides and implement in the assay and all these things. I'll not go into all the details. Essentially, we want to first test it in our this high resolution assay. Then there is a specific tag that we have to attach in the peptide that will allow it to get inside the cell. And then inside the cell, you don't want to localize it. Otherwise, you want to be localized at the periphery in the membrane fusion site. So that is we have to be take care of, and then it has to respond as a function of the stimulus. All these things has to be taken care of, and then we can say, okay, so yes, probably it is actually acting. So we are actually we have started doing some of this work. Now the reason I kind of I'll show only one thing before I finish is why we think this can work is because this similar peptide, it's not like stapled peptide, but the similar kind of peptides has been shown before to act on the fusion pore and regulate the pore properties. Here I can give you better answer how the dynamicity is controlled. This is a cytoplasmic domain of the snares. So once a pore is formed, when you added only the cytoplasmic domain, no transmembrane domain is present here. Now, if you think about it, this is my structure. This CDCIP2, which is a peptide essentially, it goes and interacts with the dynamic structure here and stops or part of the existing V and T snare interaction. Thereby, and as I said, we now see that these two are allosterically linked, this transmembrane domain and snare motifs. It probably how it actually relates how the pore property is governed there. And so it could actually close the pores, as you can see here. Then we use these peptides and put it in neurons, and we actually could see that secretion from neuron is reduced. So this thing has been done. And another paper, I was thinking about this, and this paper has come out recently. They also use this similar kind of approach where they use stapled peptide and can actually say that they're saying that they can actually show a very nice work. I mean, they could actually modulate the secretion from the epithelial cells. So that means people are also thinking in that direction, and it's not bad idea to kind of venture and go in that direction. So the bottom line is essentially this is the stage where we are actually trapping the pore and working on trying to understand the role of variety of different proteins and lipids, how it regulates the release phenomena, and can we now take it forward in different cell types, like, so for example, immune cells. It is not very well studied, but immune cell is of huge importance because you can this cytokine release from immune cell has huge implications how this, as well as say, for example, in stem cells and many other cell types. We would like to actually go and venture, and can we actually modulate secretion in those cells and so on. So yeah, I'll stop here. I'll thank all my students without whom nothing would have been possible. And then TIFR colleagues, I thank DBS members and funding sources from TIFR DA. And then, so thank you very much for listening. Happy to take questions.
Thank you, Debashish. That's very nice. So questions for Debashish, more questions for Debashish? Wait.
Yeah,
Yeah, thanks.
So once the vesicle is secreted from the cell, it is seeing the targeted cell and then it is undergo fusion. So how does this path is decided? How does that vesicle finds the targeted cell?
No,
Has to be targeted?
vesicle fuses with the plasma membrane. They releases the cargo, and then that cargo goes and they have specific receptor where they bind. There's specific proteins in the target cell where they go and bind.
No, what I understood is you have two different cells. One is secreting the vesicle, and then it is penetrating or interacting with the target cell, and then they are fusing there.
No.
No, I misunderstood.
The vesicles are present inside the cell generally,
Uh-huh.
and these vesicles contain these cargo molecules packaged inside the vesicles.
Okay.
Then these vesicles go and fuse with the plasma membrane, and then they release this cargo through that pore that I was talking about.
Outside
It's
the-
dynamics I'm
outside the
talking
cell.
about, and then they go and interact with the target cells.
And it-
Can I-
Yeah.
This, you were talking about this Parkinson's disease.
Yes.
So is it linked to excess release of the messenger particles or like excess stimulation or?
Yeah,
how
so
it is?
I tell you, this is people have been trying to understand this link for a long time. It's so complicated, this literature. What people have done is they deleted alpha synuclein, and half of the study suggests it increases the secretion, half of the study suggests it decreases the secretion. There is no concrete. So, and then what we found is that alpha synuclein protein by itself, I just showed one set of data where it showed that it is showing like a reduction, like inhibitory effect on fusion pore. But when we incorporated calcium sensor into it, which is actually the real scenario, you have synaptic vessel, you have calcium sensor also, then we did not see any effect at all of alpha synuclein. So looks like alpha synuclein's function is dependent on the regulators which actually act at the membrane fusion. We are trying to understand how those things is a little complicated.
There's a question at the end for us.
Yes, like, so what is the? What is the? Like, there is a bit of a signal. The first case is the chemical signal. But it ultimately has to have some sort of a phenomenological change, right? It's a the gene expression. So there is multiple transformations. The signal is being transformed along different steps. There are different time scales that are involved. And here we are talking about it was more diameter. So does it really transmit into changes in information as it's being processed along that multi-scale? Whatever the phenomenology is.
Yeah, it does. So if I understand your question correctly, so you're saying if the release is coupled with all other phenomenon that is happening. It has to some way or other coupled with gene expression and all other things. Like this, for example, these chemicals, when they go and interact with this target cell, like say it has receptor, specific protein where they interact, and that triggers a lot of different downstream cascade of cell signaling events. So of course it has a huge impact in this in this event. Not only that, how in the upstream also it has lot of impact.
Yeah, hi Debasis. Fantastic work. So I just have also a similar question. So when you are trying to trigger the release from the donor cell, and then you are not doing anything with the acceptor part, right? Which will eventually do the job after accepting the signal, which is coming to the vesicle. So then if I have to think about modulating some sort of physiological, if it is a pathophysiological or physiological situation, then should not I be also looking at the acceptor part? Because if I release, let's say, hundreds of cargo, which is probably naturally coming 10 cargo, and I induce it and release 100 cargo. But that is not probably going to do the job, because the acceptor also has to have some capacity, or I
Yeah.
have to enhance the capacity so it can receive, right?
Yeah, I guess I give you answer basically what exactly, Kalo asked at the very beginning, whether the amount of cargo matters for the sensitization of it does affect the sensitization. The amount of cargo release, differentially affect how the receptor molecules is accepting the pores. The answer lies in that because it differentially affect the receptor molecule, receptive cell, you actually expect that the amount is releasing would have an impact on this. Of course, I completely agree that is going to do this job. And what can happen is, it's not that there are multiple ways you can think of altering the secretion. You can think of modulating some other phenomenon, all these things. But ultimately, if I think from the secretion point of view, whatever happens upstream, it has to transmit to the secretion stage, and it has to alter the secretion. Some way or other, it has to connect with the secretory machine. That's the upstream thing. And the downstream, as I said, because we know that the amount has an impact on the receptive cell, because of that, I think this amount alteration will have significant impact on the downstream cells. Yeah.
Debasis, the chemical messenger, who is that? Is it a single molecule or
Chemical messenger is glutamate,
okay?
which is a chemical. Is a amino acid. It can be insulin, which is actually protein. It can be cytokine. It can be anything. Chemically, there is a diverse family of chemical messengers.
Okay, different families are there.
Yeah.
Okay. So one more question you mentioned, you answered Jyoti's man, that you can measure the volume or the pore size by measuring the current. Conductance. Is there any approximation, like the speed of the molecules is constant? How it is?
Speed of the molecule is generally constant.
Is it approximated or it is known?
No, it is known. It is generally constant. But here I would say, generally what people do is they do IV plot, current versus voltage plot, get a nice curve, and from the slope of that curve, they get the conductance value. And that curve is the characteristic of what you are mentioning. If there is any issue in terms of the non-linearity, it should be reflected there. When you change the voltage and measure the curve. That is one thing. In this case, it is true. But yeah.
Okay. This one question.
Since you mentioned that this monk actually regulates the size of the pore as well, so when you are designing these peptides, and if you change the KD value of these peptides, and since they interact with NUNC1, will it also be able to regulate the size of this pore to different sizes? And does the pore of the size also determines the specificity of the cargo?
So if I understand correctly, you are asking whether the peptides interaction determines the cargo's.
No, so there are like two questions. So first, the NUNC1 actually regulates the size of the pore, right? So since you are now designing different peptides, so if you change the KD value of these different peptides, say 1 to 13, okay? So the dissociation constant is going to change, and that will also lead to change in the interaction that is happening.
That can change the pore properties.
Yeah, that can change the pore properties.
Absolutely.
And that can in turn change the cargo specificity, the kind of cargo that is being transported through this pore.
That I don't know.
Okay.
That I don't know unless you do the experiment.
Okay. Thank you.
That's it.
Okay, well, those are some wonderful questions, and I think we ended right on time. So I'll invite every one of us to, first of all, thank Debasis and then walk with us outside the West Canteen for some snacks. So thank you very much. Thank you, Debasis. And thank you, auditorium staff. Thank you very much.